Well, Ryan, I'm not sure I'm going to be able to follow up with that fantastic introduction. I would like to start by just thanking all of you to be here tonight. This is a great honor, and I am humbled to be among those amazing investigators who stood here before me to give the Kligman-Frost lecture. Now, my message tonight is both scientific and personal. Scientifically, my research program has over the years studied fundamental mechanisms that support multicellularity. And in order for tissues to become multicellular, let's see if I can avoid some of the previous, okay. In order for tissues to become multicellular, they must be able to connect, to communicate, and to cooperate. And these are precisely the attributes that we as a scientific community really need to do to be at our best. And that is my personal message tonight, especially to those of you just starting out in your careers. So, how did multicellularity arise during evolution? Well, the evolution of cell-cell connections was absolutely essential. Without cell-cell connections, interfering with these connections leads to human diseases, particularly in tissues that experience a lot of mechanical stress, such as the skin and heart. Now, if we go back in evolution to ask when is the first appearance of cell-cell connections, we actually need to go all the way back to single-celled organisms called choanoflagellates. Now, what were primitive adhesion molecules doing in choanoflagellates? We really don't know for sure, but it's been speculated that since they were concentrated in the feeding organ of the organism, that they might have been involved in capturing prey. It wasn't until later in sponges and in organisms such as Hydra that were forming simple epithelial sheets that we actually see these primitive adhesion molecules called cadherins co-opted to assemble into primitive Adherens junctions. And then it wasn't until we see vertebrates evolve that these very complicated and highly organized specialized junctions called desmosomes first appeared. So I wanted to put desmosomes in the context of the other types of intercellular and cell substrate junctions that we know about, just to give some background for our discussion tonight. So desmosomes are one of 4 different types of intercellular junctions. Tight junctions, which are important for establishing polarized epithelial cell sheets. They form a barrier and they regulate the passage of molecules between cells and also through cells. And then we have gap junctions. Gap junctions form pores in the plasma membrane that allow for direct transport of small molecules, ions, nucleotides, and also facilitate the electrical coupling of cells, which is very important in cardiac biology and also in the skin. And then we have the Adherens junctions and the desmosomes, which are very similar in certain ways. They both attach different elements of the cytoskeleton to the plasma membrane. In the case of Adherens junctions, this is the actin cytoskeleton, and the Adherens junctions organize the contractile actin machinery next to the plasma membrane. And in the case of desmosomes, they anchor the intermediate filament cytoskeleton to the plasma membrane, and in so doing, they form a scaffolding throughout the entire tissue that helps to resist the forces of mechanical stress. Desmosomes are particularly prominent in stratified complex epithelial tissues, such as the epidermis. And if we look at an electron micrograph of a desmosome from one of these complex tissues, we can see that they're highly organized, they're very complex. I think they're quite beautiful, actually, and their structural complexity is mirrored by their molecular complexity. And so in order to simplify this complexity, we can actually describe the desmosome as being made up of 3 different protein modules. The first of which is the adhesive core of the structure, and this is made of cadherins. And in desmosomes, these cadherins can be subdivided into 2 classes, desmoglians, which many of us know as pemphigus antigens, and desmocollins. And then the second module is the armadillo family. And in desmosomes, these contain placoglobin and placophilins, and I think most of us know that placoglobin is very similar to beta-catenin, and these are known to be important, both in junctions and also in nuclear functions such as transcription and other functions that are just now emerging. And then the final module is the cytoskeletal anchoring module. And in desmosomes, this role is performed primarily by desmoplakin, which is part of a protein family called the plakin family. And desmoplakin anchors intermediate filaments through its C-terminus, and then it associates with the plasma membrane complex of proteins through its amino terminus. But it's even more complex than this because adhesion takes on new dimensions in stratified epithelia. And in fact, there are 7 different desmosomal coherent genes that are expressed in human epidermis, and these give rise to at least 10 protein isoforms. So why are there so many of these adhesion molecules? It leads to the question of why the complexity. If these molecules are simply performing adhesive and structural functions, why do we need so many of them? This is a question that my laboratory, that drives it actually on a daily basis, and it's also a question that's being asked by a number of us in the field. And I would like to address the hypothesis tonight that desmosome diversity, which was selected for during the evolution of vertebrates, contributes to the functional complexity of vertebrate tissues. So it affords the opportunity for vertebrate tissues to have additional functions. And a corollary of this hypothesis is that these desmosomal coherens and the other desmosomal molecules are playing functions that transcend their role in adhesion. So I thought I'd take you on a little walk through memory lane, as some people do during talks like this. And actually, a retrospective analysis of my whole career path revealed to me, as I was preparing for this talk, that I was actually interested in desmosomes many years ago in college at Pomona College when I was taking a cell biology course. And we were asked to draw all of the intercellular junctions that we learned about in class at a molecular level of resolution. Well, of course, we didn't know what any of the molecules were. So the best thing I could do is go back to the Singer and Nicholson paper that was published in 1972 proposing the fluid mosaic model of membrane organization. And as you can see here, my picture of the desmosome shows that bias. But what I really want to point out to you is this arrow, which is pointing to a few of these potato-like molecules on the inside of the plasma membrane. And I labeled at that point as the substance of unknown chemical nature. So even then, I was intrigued with what proteins were attaching the cytoskeleton to the plasma membrane at the desmosome. And a few years later, I was still interested in that question, and I was fortunate to be able to tackle it when I went to Bob Goldman's lab. Now, Bob is the chair of the cell biology department, still at Northwestern University, and I met him at Woods Hole at the physiology course at the marine biology lab during the summer, and he invited me to come to his lab. Bob is a pioneer in the study of intermediate filaments and intermediate filament functions and was one of the first to identify vimentin. So I packed my bags and I went to Chicago to do postdoctoral work, and I was also introduced to this group of people, and some people not shown here, who are experts in intermediate filament biology: Peter Steinert, Jonathan Jones. For those of you who know my husband, Rex Chisholm, who will make an honorary intermediate filament biologist just for tonight, and also people like Dennis Roop and others not shown here. Also, when I got to Chicago, I met another group of very important people in my career, and those were the cutaneous biologists and June Robinson and Amy Powler and an expanding group of people at Northwestern interested in skin biology. And these folks got me to join the Society for Investigative Dermatology. In fact, Amy was my roommate at my first meeting and shepherded me around the meeting. And this was a really critical point in my career because I've really benefited from this fantastic group of people. So, back to the question of what's connecting those filaments to the membrane, a question that I addressed later in my time in Bob's laboratory. So another very important contributor to this question were the residents of the stockyards in Chicago, which sadly don't exist anymore, but these bovine friends contributed, unfortunately, much of their tissue in order for us to be able to isolate desmosomes. And so we would isolate desmosomes, enrich for the proteins in desmosomes, and these proteins, in particular, these high molecular weight proteins up here, based on work done in Werner Franke's laboratory, we predicted would be the intermediate filament binding proteins. So to make a long story short, we used expression cloning to isolate clones, which turned out to be desmoplakin. Over many years, we patched together a 9.5 kilobase cDNA. We sequenced from many sequencing gels by hand, and we came up with this protein model of desmoplakin structure in collaboration with David Perry, who's a biophysicist in New Zealand, and came up with a model that depicted desmoplakin as a 3-domain structure with a central coiled rod domain and 2 globular end domains. And the sequence motifs in the C-terminus suggested to us that it might be an intermediate filament binding protein. And in fact, that turned out to be correct. There was also a very interesting repeating motif in that domain, which John Stanley, who was looking at the bullous pemphigoid BP230 kilodalton protein at the time, found the same motif in that protein. And thus, a new protein family emerged, which is now called the plakin family, which is a coin termed by Fiona Watt. And that protein family has grown. Fast forward to 2001, and my laboratory worked with Bill Weiss to determine the high-resolution X-ray crystallography structure of the first intermediate filament binding domain, and that was the C-terminal domain of desmoplakin. So all of that structural work on the desmoplakin protein laid an essential groundwork for our functional studies and ushered in, as Ryan Hobbs, who I assume is in the audience, would say, a decade of research on desmoplakin. And so this crew right here was the first crew who began to study desmoplakin function and the importance of the intermediate filament connection to desmosomes. And we generated mutants that abrogated or interfered with the desmoplakin intermediate filament connection by deleting that the intermediate filament binding domain. And I want to show you some movies, and Robert, if you can just start these, both of these movies here, which are older movies that were published by Arthur Hewen in the laboratory. And they compare control desmoplakin with keratinocytes expressing the mutant desmoplakin. And I think you can see very clearly that that disrupting the intermediate filament connection interferes with the integrity of the entire keratinocyte cell sheet. So desmoplakin intermediate filament interactions are clearly very important for this tissue integrity. And now fast forward to today, a number of collaborators and colleagues have identified mutations throughout the desmoplakin molecule, including those that delete portions of the C-terminal IF binding domain. These lead to human diseases such as Carvajal syndrome, in which patients have skin defects, woolly hair, and a form of cardiac cardiomyopathy called arrhythmogenic cardiomyopathy that leads to sudden death, and also very lethal forms of desmoplakin deficiency, such as lethal acantholytic epidermal isobulosa. But why do desmoplakin mutants misbehave? Is it simply defects in the mature form of the desmosome and a loss of the tissue integrity that causes these diseases, or are there other reasons? And I'd like to give you some examples of some other reasons my laboratory has uncovered for the pathogenesis in these diseases. And one of them has to do with the dynamics of desmoplakin and interference with that dynamics during remodeling of epithelial tissues. Lisa Godsell and Eva Margot in the lab took advantage of green fluorescent protein, and they made a desmoplakin, fused the green fluorescent protein, introduced that into keratinocytes, and using optical imaging methods, looked at the dynamics of desmosome assembly and desmoplakin dynamics in particular during epithelial remodeling. And they found in a paper, nice paper published by Lisa in the mid-2000s, some very interesting features of desmoplakin dynamics that are going to tell us a lot about human disease. I'll show you in a bit. And I'm just going to play, if you could play this movie, Robert, I will talk you through what we're seeing here. And if you look at this cartoon on the right, this summarizes the different phases of desmoplakin dynamics during assembly. So in an initial phase, and you'll see this movie loop through, so you'll see it again, we see very rapid accumulation of these little puncta of desmoplakin during the first few minutes after cell contact. And after that, we see a whole Milky Way of spots of desmoplakin puncta that appear in the cytoplasm near the forming borders. and subsequently translocate to the forming desmosomes, which are strengthening in their adhesion. That translocation, we now know, is due to changes in actin organization. These are not microtubule-dependent processes. These are not vesicles. They're actually non-membrane bound macromolecular complexes. And these types of movies actually solved or addressed a controversy that had been in the field for a while as to whether the spots that were seen in fixed tissues or fixed cells were remnants of old desmosomes or actually could serve as precursors. And clearly, they can serve as precursors. Now, this dynamics is very important for the maturation of desmosomes and for the acquisition of strength within a tissue. And so, Lauren Albrecht, when she came to the laboratory, was very interested in what regulates these dynamics. We had known for a number of years, down here, we had known for a number of years that there was a serine site at 2849 that was extremely important for regulating this process. But what Lauren did was she actually did a comprehensive mass spectrometry analysis in collaboration with Don Hunt at the University of Virginia, and she found that this was not such a simple thing, but in fact this is a priming site for processive phosphorylation through GSK3 beta kinase. And in the next slide, if you can start the movie on the right, well, you can have both movies going at the same time if you can. So what happens if you mutate this priming site, then you abrogate the entire processive phosphorylation cascade, and that leads to this dramatic change in the dynamics of desmoplakin to a form that's all stuck along intermediate filaments. So it's a tight binding form. So this is a quite dramatic, and actually if you do quantitative assays, desmoplakin binds to intermediate filaments an order of magnitude more tightly if you interfere with this cascade of phosphorylation events. Now, Lauren became even more interested in this because it was discovered that there's a mutation in this region in arginine 2834. It changes to a histidine in cardiocutaneous disease. And in fact, we made a transgenic mouse, or actually we collaborated with Jeff Tobin's lab to make a mouse that expressed this in heart, and that led to the mice having basically arrhythmogenic cardiomyopathy. So Lauren made cell lines, and she put this desmoplakin into keratinocytes and saw that it turned into a tight binding sticky form of desmoplakin, just like the phosphodeficient mutants I showed you. Now, how is that happening? So this is at an arginine, it is not a serine, and through mass spectrometry, we found that it's methylated, and that mutation abrogates methylation, and furthermore, loss of methylation prevents the processive phosphorylation. So it appears that a priming serine and a methylation site are cooperating to promote processive phosphorylation, and that regulates desmoplakin dynamics, and mutation that leads to human disease can interfere with that molecular process. So how does that all affect desmoplakin? How do these post-translational modifications affect desmoplakin? Well, we're still trying to figure that out, but Lauren's idea is that this region of desmoplakin is acting as a signaling hub that actually recruits and binds to the enzymes that modify its post-translational modifications, and that if you don't have the right modifications, it releases those enzymes into the surrounding microenvironment, and that can affect global signaling. So that's what we're working on now. And so I just wanted to show you all the other people working on desmoplakin in the past and currently that I don't have time to talk about, but Amanda and Ryan are both in the audience. Josh will be talking on Saturday, and Chen, future talks, I'll tell you about Chen. So I've been telling you a lot about the C-terminus of desmoplakin, but the amino terminus is also very interesting, and Deepal Patel in the lab, who is an MD PhD student now back in the clinics, was interested in some mutations in that region of desmoplakin. And these mutations cause both skin and heart disease. And so she hypothesized that because the mutation hotspots in the immunoterminus are downstream, they're away from the binding sites for the known armadillo protein binding partners, that there probably are other partnerships that are important in this region. And so she carried out a yeast-2 hybrid screen and found a new binding partner called N-binding protein 1. This is a protein that binds to the plus ends of microtubules. And we were really interested in this because Terry Leckler had previously shown that desmoplakin is important for microtubule organization, but through association with minus N binding proteins. So Diepl showed that this EB1 molecule and the plus tips of microtubules impinged upon desmoplakin-containing desmosomes at right angles. And what happens, and also she showed experimentally that desmoplakin stabilizes these microtubules and acts as a cortical capture site to do that. So if you can start these two movies simultaneously, I'll walk you through what happens in control cells with microtubules versus desmoplakin deficient cells. So in control cells, you can see these plus tips of microtubules impinging at right angles, and they become stable here. But in desmoplakin deficient cells, the microtubules behave very differently. They come in at acute angles, and they don't find their cortical capture sites, and they're not as stable. So why is this important? Well, it turns out that EB1 had been implicated in the trafficking of connexins. Connexins are gap junction assembly subunits, and they're important both for electrical coupling in heart cells, and they're important, as we all know, for epidermis as well, the two tissues that are affected in desmoplakinopathies. So Diepl looked at both connexin trafficking, and I'm not showing you that data here, but I'm showing you some functional data of wild-type desmoplakin, as well as a series of cells that are expressing mutants, some of which are unable to bind to EB1 because of the mutation. And what this is a dye transfer assay in which you inject the yellow dye into one cell and then watch it to move through gap junctions into the neighboring cell. And you can see that wild type can transfer the dye very well through gap junctions, but three of these, this one, and this one, which have abnormally localized EB1, cannot transfer dye through the gap junctions, indicating that desmoplakin and EB1 interactions are important for gap junction function. So then this Diepl's work then tells us that EB1 binding to the amino terminus of desmoplakin in a region that's mutated in cardiocutaneous disease, if ablated in those diseases, affects the assembly of gap junctions and gap junction function. So again, this is extremely important, we think, has implications for both heart and skin disease. So now in the last few minutes, I'm going to move back to the complexity of desmosomes in stratified tissue. And as I mentioned before, desmosomes, desmosomal adherens come in multiple, 10 different proteins are expressed throughout the epidermis, and they're in a highly patterned distribution. And what is the meaning of this? So I'm going to tell you about our work on one of these desmosomal adherens called desmoglein-1, which is first expressed as cells are committing to differentiate, and then it gets concentrated in the basal layer. And we know that ablation of desmoglein-1 function from a variety of ways leads to various types of diseases, including the autoimmune disease pemphigus foliaceus, genetic diseases such as stripe palmoplantar keratoderma, and the recently described metabolic wasting syndrome from Ellie Sprecher and his colleagues, and also toxin-mediated bullous impetigo. So it's a really important disease target. And a number of investigators over the years, including Andrew Kowalczyk when he was in the laboratory, and our longtime collaborator and friend Masa Amagai, were interested in understanding the adhesive function and also as a pemphigus target, up to current times when Ellie Sprecher worked with a group of people to identify the importance of desmoglein in genetic disease. But what I want to tell you about is the role of desmoglein in normal epidermal morphogenesis. And this question was addressed by this crew of people here, starting with Spiro Getzios and Corey Simpson, also here tonight, I think, and then Bob Harmon, Linda, and Jen, who utilized 3D organotypic models, which many of us are familiar with, to address the function of desmoglein-1 in epidermal morphogenesis. So they, and actually you can see here in control cells how nice this model replicates the features of normal epidermis, both structurally and also in the biochemical program of differentiation, where the suprabasal markers are expressed very nicely. However, if one ablates desmoglein, both the biochemical program of differentiation, we can see here suprabasal keratins are no longer expressed normally, and the structural features, including keratohyalin granules, are not properly assembled. So we concluded that desmoglein guides differentiation. It's not just a byproduct or a result of differentiation, it actually promotes the differentiation process. And this it also is important for regulating the cell shape and size through, we think, an actin remodeling process. So this leads to two different arms of a model that suggests desmoglein both is important for driving a biochemical program of differentiation as well as a morphological cytoskeleton-dependent program of differentiation. So then Bob Harmon got into the game here, and he was very interested in expanding on the signaling pathway that desmoglein regulated, which was revealed by Sparrow and Corey's work. They had earlier demonstrated that desmoglein attenuates the MAP kinase signaling pathway through EGFR and also RAS, RAF, and preventing or attenuating MAP kinase signaling. So Bob wanted to know how this happened. So he carried out a yeast-2 hybrid screen and found this protein here called urban, and then demonstrated in a very nice series of results published in the Journal of Clinical Investigation that urban and its binding partner, a scaffolding protein called SHOC2, prevents RAS and RAF from coming together, and this is what attenuates that pathway to promote differentiation. But what about the other arm, the actin remodeling arm? Well, this is a work in progress that is being addressed by Oksana Nekrasova in the laboratory, and she also looked at the hits in the yeast-2 hybrid screen that Bob Harmon carried out and found something that she thought could be a good candidate for mediating these cell shape changes. And it's called TC-TEX, which is a dynein light chain. So it's part of a molecular motor that moves vesicles to the minus ends of microtubules. But it's also been indicated as regulating actin. So it has two functions. And what Oksana did is she identified the TC-TEX binding site in desmoglein and then made a desmoglein that can't bind to TC-TEX in order to address its function. And she was able to show that indeed a TC-TEX binding deficient desmoglein is unable to concentrate and remodel the actin cytoskeleton in the cortex of differentiating keratinocytes. And another thing she showed is that the normal segregation of desmoglein and E-cadherin doesn't occur properly. So she is hypothesizing based on this that TC-TEX gets desmoglein to the proper domain. It segregates it into the proper domain on the plasma membrane. And that in doing so, it puts it in position to regulate the cortical actin cytoskeleton. Well, how does it do that? That's a question she's addressing, but first she wanted to see, well, is TC-TEX important in epidermal morphogenesis? No one's ever looked at TC-TEX in the skin before. It turns out, using this organotypic model, that TC-TEX is important for giving the normal stratified layers of epithelium, and we're addressing whether or not this regulation of cortical actin is involved in giving it that phenotype. So finally then, the model that Oksana has is that desmoglein binding to TC-TEX gets it into the proper place on the plasma membrane so that it can perform its function of actin remodeling. And she has furthermore showed in experiments that I don't have time to show you, this requires the dynein motor and another binding partner called cortactin, which is actually an known actin binding partner. So we're in the process of figuring out how this molecular machine regulates the cell shape of keratinocytes that are differentiating. So this is a crew that's also been working on other aspects of desmoglein function that I don't have time to tell you about today, but listen for their talks in upcoming meetings. Some of them will be going to the Epithelial Gordon Conference, and Jody Johnson will be speaking tomorrow, I believe, in a special cutaneous surgery talk, so you'll be able to hear about her work there. So my final concluding science slide is that I hope I've shown you that desmosomal diversity going beyond its adhesive function is very important for the functions of differentiating keratinocytes to make vertebrate epidermis. And so finally, back to the personal message. I'd just like to thank all of the people that internationally have been on my team and have supported my career throughout the years. One of the best things about science is the ability to interact with people all over the world, and this leads to great connections. Great connections make great science. But great connections also make great friends all around the world. And I'm showing just a few of the pictures that I've accumulated over the last 30-some years that Rai was telling about, and many more could be put up here. So I'd love to show my photo album over drinks if anyone wants to see it. And I'd also finally like to thank all the members of my research laboratory over the years. Those in red or orange here are my current lab members. I would not be here without them. They really are what makes the lab run. And again, all of the past and present lab members throughout the years whose shoulders I am standing on and who help everything stick together. It's really important. So with that, I'd like to finally thank another really important group of people, and those are the people who fund this work, especially for this work that I presented today, NIAMS, the Derm Foundation, also the Northwestern Skin Disease Research Center. So with that, thank you very much for your patience and for all of your support.